And now we're going to start chapter 10, which is all about the blood. We're actually going to cover a few chapters that span the cardiovascular system. The first topic in our cardiovascular system is blood. So we'll look at blood now. So let's talk about what blood is. If you recall back from chapter 3, blood is actually a connective tissue. It's a liquid connective tissue. The formed elements are the other name for cells that are within the blood. And there's different types of cells that we'll talk about. Of course, you know this. There's lots of plasma within your blood. That's the liquid matrix that surrounds our formed elements. Some of the characteristics: it's thicker than water. You've heard of that before, right? Blood is thicker than water. Temperature around 37 degrees Celsius. That's body temperature, 98.6 degrees Fahrenheit. The pH of blood is always kept somewhere between 7.35 and 7.45. It's about 8% of your body weight, which most of us probably don't think about how much our blood actually weighs. So if you weigh 200 pounds, 16 pounds of you is just blood, which is kind of interesting. Average circulating volume differs for males and females because males tend to be larger, 5 to 6 liters for male, 4 to 5 liters for female. The difference, of course, is due in that body size. When you donate blood, you're generally donating about a liter of blood. So let's talk about some of the important functions of blood. There's 3 important functions of blood. One is transportation. Transportation is the delivering of oxygen from the lungs to the body cells, and then the delivering of carbon dioxide from the body cells to the lungs. It's also going to transport nutrients as well as waste products and hormones. It also helps with regulation, helps maintain a stable body pH of 7.35 to 7.45. It helps to distribute heat to maintain a stable body temperature throughout our body. It also helps to influence the water content within our cells and tissues through the use of osmotic pressure. It also helps to protect our body. It forms clots after an injury. When the blood vessels are damaged, it's going to clot those vessels to prevent blood loss and maintain cardiovascular function. It also contains special immune system components, our white blood cells, to help protect against invading organisms and disease. These include antibodies, antimicrobial proteins, and the white blood cells, as I mentioned. So the plasma or liquid portion of our blood is the majority of blood volume. 55% of blood volume is just the liquid part. It's made of mostly water. There's also proteins within it. Those proteins are made mostly by the liver. They include albumins, globulins, and fibrinogens. They all have special roles. There's also solutes within our blood, including salts and electrolytes, nutrients, waste products like carbon dioxide, hormones, as well as gases would all be dissolved in our blood. As I mentioned, the majority of our plasma is water. Over 90% of the plasma is water, and that serves to act as a solvent for carrying the other substances, as well as to help absorb heat so that our body maintains a stable temperature. Proteins are also within our blood, as I mentioned, the albumin and globulins. They help with pH buffering, osmotic balance, as well as blood clotting, helping to transport antibodies and other lipids throughout our body. Some of the other solutes, including sodium, potassium, and chloride, they help to maintain the osmotic balance as well as pH and help to regulate the permeability of our cell membranes. Some of the nutrients include glucose and fatty acids coming to and from tissues for metabolic purposes. Some gases, such as oxygen, carbon dioxide, and nitrogen, are dissolved within our blood. There's also regulatory substances like hormones, which can be bound to transport proteins or free-floating. And then waste products like urea and uric acid, which come from the breakdown of proteins. So, some of the formed elements or cells or cell components in our blood include what you see here on this chart. There's the white blood cells, which are ultimately involved in fighting off invading microbes. They also help to eliminate cell debris. They make up a very small percentage of the formed elements. There's also platelets. Platelets are cell fragments. They're not actually cells themselves. They're kind of little pieces of cells. They aid in blood clotting, although they make up less than 1% of all of the formed elements. The majority of the formed elements in our blood are the red blood cells. They're disc-shaped. They have a protein called hemoglobin, and they transport the oxygen and carbon dioxide to and from the tissues. So let's talk about these red blood cells, the major component within our blood. Their technical term is erythrocytes, that suffix "cyte" referring to cell. Erythro is the Latin word for red, so red cell is what they are. 7 to 8 micrometers in diameter, so fairly small when you compare them to other cells. They're biconcave, so they have a disc shape, providing a large surface area for gas exchange with the carbon dioxide and oxygen. There's no nucleus, which allows for that biconcave shape, and there's very few organelles. Its main job, once these red blood cells are made, is to just float around the blood carrying the oxygen and carbon dioxide. They have a very flexible membrane to allow maneuvering through the blood vessels, so they can kind of squeeze and squish through the small capillaries. And as I mentioned, they contain the hemoglobin protein, which helps it carry the oxygen and carbon dioxide. Hemoglobin is that iron-based protein that helps to bind to that oxygen and carbon dioxide. You can see here the overall sectioned view here where it's biconcave, caving in on both sides. The hemoglobin molecule itself here contains iron. That iron is going to help increase the binding affinity of oxygen. So in other words, it's going to make this molecule act like a magnet for oxygen and hold lots and lots of oxygen as it is transporting throughout the blood. It also gives red blood cells its reddish appearance. When it's saturated with oxygen, those hemoglobin molecules make our blood that deep red color. Platelets are the tiny cell fragments of larger cells called megakaryocytes. Megakaryocytes are found within the red bone marrow. Basically, a small amount of cytoplasm is enclosed in a little portion of cell membrane, and that's what makes up a platelet. It's important for blood clotting. They cannot reproduce because they're only cell fragments. You can see them here in this image. They're these little teeny little specks that you see scattered amongst the larger cells. Of course, the majority of the cells that you see here are the red blood cells, and then these large purple cells which have little blobs inside of them. Those blobs are the nucleus of those white blood cells. And you can see there's different types of white blood cells. We'll look at those in a minute. So white blood cells in general are known as leukocytes. That suffix "cyte," leuko, is the prefix meaning white, so these are white cells. These guys are their main role is to protect the body. They engulf invading bacteria and viruses, kind of like Pac-Man. They can also release chemicals to initiate other immune responses, and they can produce antibodies to help forge an attack on an invader. There's 5 types of white blood cells: neutrophils, lymphocytes, monocytes, eosinophils, and basophils. So all of the white blood cells have a nuclei. They can be granular. In this case, the granular white blood cells include eosinophils, basophils, and neutrophils. They're called granular because when you look at them under the microscope, they appear very, very grainy. Then you have these agranular white blood cells, which are the lymphocytes, which are very small, compared to the monocytes, which are very, very big. They have just a single nucleus, and you don't see all of those grains inside of them. So hemopoesis is the technical term for blood cell production. It's also known as hematopoiesis. This occurs first in the yolk sac of the embryo, and then within the liver, the thymus, the lymph nodes before birth. In adulthood and about 3 months before birth, it begins to occur in the red bone marrow. Of course, embryos, when they're very early in development, don't have bones, so they don't have the red bone marrow for where hematopoiesis is normally occurring. So in adults, right now, your red bone marrow is creating your blood cells. Your bone marrow cells are called pluripotent stem cells. These pluripotent stem cells can differentiate into all of the other types of cells that you need within your blood. So here you can see the pluripotent stem cell, which lives within your bone marrow. It can be stimulated by different hormones and be told to create eosinophils or mast cells or megakaryocytes or retinaculocytes, which will become red blood cells, or monocytes, etc., etc. Erythropoiesis is the technical term for red blood cell formation. The reticulocyte is the pre-red blood cell. The reticulocyte basically pops its nucleus out, and then it forms that biconcave erythrocyte. Mature red blood cells cannot reproduce because they don't have a nucleus, so they die within about 120 days. The stimulus for reproduction of red blood cells is reduced oxygen delivery to tissues, which is also known as tissue hypoxia. The kidneys are detecting the hypoxia. They release a hormone called erythropoietin. The erythropoietin communicates to the red bone marrow that it needs to produce more reticulocytes and more red blood cells. Here you can see how the red blood cell formation is regulated. It's again stimulated by some type of disruption in homeostasis, decreasing oxygen delivery to the kidneys as well as other tissues. So those kidney receptors detect those low oxygen levels, send information to the red bone marrow to form more reticulocytes. The reticulocytes enter the bloodstream, eject their nucleus. Now you have lots and lots of red blood cells, so you have increased oxygen levels, and then you get that negative feedback loop going where the kidneys are then turned off from secreting the erythropoietin. Leukocytosis, this is when the lymphocytes proliferate in the lymphoid tissues through mitosis. So leukocytosis is going to increase the white blood cell quantity. This is in response to infections, exercise, anesthesia, or surgery. Rate of red blood cell production is equal to the rate of red blood cell destruction, except in cases of hypoxia where red blood cell production is increased. Worn out red blood cells are dismantled in the spleen. The spleen is kind of like the red blood cell graveyard, as well as the liver and red bone marrow. That hemoglobin is broken down and reused and recycled. The iron is recycled to red bone marrow and put back into new red blood cells. The non-iron portion of the heme are broken down, processed by the liver, and excreted by the kidneys and intestines. The globin portion degrades into amino acids and is then released into the bloodstream and reused. So as your red blood cells are dying, they go to the spleen, and then some of those hemoglobin counterparts taken to the kidneys for excretion, and then others are reused in new red blood cells. So this diagram shows you the formation and destruction of red blood cells. So you have your red blood cells circulating for about 120 days or so. Macrophages within the spleen and liver, red bone marrow are going to phagocytize those worn out red blood cells, break down the hemoglobin into the heme and globin components, which you see here in 2 and 2A. The iron is removed from the heme. The iron binds to a transport protein called transferrin. This transferrin protein travels to the bone marrow, where it's then taken up by new reticulocytes and reused in new red blood cells. The globin protein, where'd my mouse go? Here it is. Globin protein is broken down into its amino acids, where it's released into the blood, and then those are used to make new proteins in the blood. They can also break down into something called biliverdin, a green pigment. That biliverdin is broken down to bilirubin, which is a yellow pigment. That bilirubin travels to the liver and is used to create the bile that goes and is secreted into the small intestines. So there's two kind of pathways here that take place with the breakdown of our red blood cells. So you have the heme breaking down to the iron and the non-iron part, and the globin breaking down to the amino acids, which are reused. The non-iron part of the heme is turned into bilirubin, turned into bile, and secreted in the feces. The iron component is transported to transferrin, which goes to the red bone marrow to be used with erythropoiesis. Hemostasis is a series of physiological responses that's used when your blood vessels are injured or damaged. So first you have a hemorrhage, which is a large amount of blood loss. Then you get a vascular spasm or vasospasm that's going to restrict the blood flow into the damaged vessel. So in other words, reducing blood loss. Then a platelet plug will form, sealing the damaged vessel. Then a blood clot and coagulation forms. This is a highly complex process that ultimately seals the damaged area and allows for complete tissue repair deep to that superficial damage. After the blood clot seals, the platelets inside the clot pull on fibrin threads to contract the clot. Contracting the clot is going to remove excess fluid. It's going to bring the damaged surfaces close together for permanent healing, and then those clots are dissolved from the inside out in a process called fibrinolysis. Fibrinolysis is within the formation of the clot. A protein called plasminogen is incorporated. Plasminogen is converted into plasmin in the presence of blood and other tissue factors such as thrombin, factor 12, and tissue plasminogen activator. That plasmin digests the fibrin, dissolves the clot once the damage is repaired, which is why, and you've probably noticed this if you've ever had a cut that required a scab, the scab almost falls off towards the end, and that's because the skin has completely healed underneath. All of the fibrinogen and fibrin have formed together, forming that scar tissue, and that clot is no longer needed. A thrombus is a small clot formed of unbroken blood vessels, and it usually dissolves spontaneously. If it remains, it can kind of flow through the circulatory pathway and interfere with blood flow to various organs. An embolus is a clot that's broken off and travels through the cardiovascular system. It eventually blocks small blood vessels. It can lead to pulmonary embolism, heart attack, stroke, or even kidney failure. Those emboli can be dissolved by injecting tissue plasminogen activator, which dissolves the clots and breaks them down. If it's quickly treated, damage to the blood vessels can be minimized. So this is hemostasis in a little diagram here. So when you have damage to your blood vessel, immediately those vessels are going to spaz, which is going to decrease the blood flow to that area. That can last for minutes to hours depending on how big that damage is. Within seconds, you have your platelet plug formation. So all of those platelets that are scooping by kind of stick to that opening. Those platelets secrete special chemicals to activate other platelets, and those activated platelets stick together, blocking blood flow in that vessel. Then the last step is the clotting and forming a blood clot. This requires an enzyme called prothrombinase. Prothrombinase and calcium are going to convert prothrombin to thrombin. Thrombin is going to convert fibrinogen to fibrin, and fibrin forms threads, which traps blood cells and forms the blood clot, which once it dries out, it forms the scab. Let's talk about the difference between antigens and antibodies. An antigen is a substance that can provoke an immune response. It's a protein on the outside of the cell membrane. This is ultimately what your body uses to recognize invaders. All of your body cells have a little label on them that say, "Hi, I belong here. Hi, I belong here." But invading bacteria cells have antigens that say, "Hi, I'm a bacteria cell. I'm trying to kill you." So your immune system cells go around detecting foreign antigens in your body. An antibody is a protein that's found in blood that binds to specific antigens. So for instance, if you've had a flu shot, you now have those flu antibodies floating around in your blood, and when your body comes in contact with the flu, your antibodies can respond. When antigens and antibodies come in contact, a reaction occurs. It's called agglutination or clumping. That is ultimately going to cause hemolysis or the bursting of the cell. So, this is related to blood types. There are 3 types of antigens, or really 2 types of antigens that occur on our blood cells, and that would be A antigens or B antigens. If you have blood type O, that just means you have no surface antigens on the outside of your red blood cells. So the antibodies that are produced in our blood in response to blood types are anti-A antibodies or anti-B antibodies. There's also an R group. There's an antigen that's RH, and the antibody is the anti-RH. So let me try to do one of my crappy whiteboard drawings for you to kind of show you what I mean in this case. So, if you're blood type A, that means your red blood cell, there's a red blood cell, has little A antigens all over its surface. If you're blood type B, this means that your red blood cell has B antigens all over its surface. If you're blood type AB, guess what that means? You have both A and B antigens all over the surface of your red blood cell. And if you're blood type O, what does that mean? that means you have no antigens on your red blood cells. There's also this Rh factor, this Rh group. You can be Rh positive or Rh negative. So, what that means is you can have Rh positive or Rh factors on your blood. In that case, this person's blood would be A positive. This person's blood would be B positive. This person's blood would be AB negative because they do not have that positive Rh factor. This person's blood would be O negative. Now, when people that have A positive blood come into contact with, or I should say when A positive blood comes into contact with B positive blood, they're going to agglutinate or clump together. They're not going to fuse nicely. The people that have A blood produce B antibodies. In other words, people that have A blood cannot receive blood from type B donors. Their blood will clump and it will cause a serious overall reaction leading to systemic failure. However, people that have A B blood can receive blood from people that are type B or people that are type A. In this case, though, this person's blood is AB negative, so they cannot receive blood from this person who's B positive or blood from this person who's A positive, because these people have that Rh positive antigen. So this person's blood is going to respond to that antigen and reject that blood. This person that's AB negative can only receive blood from this O negative person in this instance, because this O negative person does not have any antigens on their red blood cells. So when these red blood cells mix with these red blood cells, they're not introducing anything new. They're not introducing an Rh factor or an A or a B. They're just introducing empty red blood cells, which means the AB negative and O negative bloods will mix fine. However, if this O negative person needed a donor, just plug it in my computer here, sorry. If this O negative person needed a donor, they would not be able to get blood from this AB negative person because this AB negative person has A and B antigens. This O negative person's blood will respond and attack those A and B antigens. This O negative person also cannot receive from this B positive or this A positive, again, because he's receiving antigens that he doesn't normally have on his blood. So unfortunately, while this O negative person can donate blood to this guy, or this guy, this O negative person can only receive blood from other people that are O negative. Which is why if your blood type is O negative and the Red Cross knows about it, they're probably calling you on a very frequent basis asking you to donate. So this is what happens in a blood typing test. You have 4 samples of blood in this case. This blood here is blood type A. When you treat it with anti-A serum, it clumps. When you treat it with anti-B serum, it does not clump. So that tells you that this is A blood. In this case, you have clumping in the B. So this tells you that they have the B antigens. In this case, you have clumping in the A and B. So you see that they have both A and B antigens. This person here's blood does not clump for any of those tests, so that you know it does not have any of the antigens, and therefore it has to be type O blood. Here you can see that compatibility that I was referring to. People that are blood type A can donate or can receive blood from other A's and other O's and O blood. They're incompatible with A and AB. So in other words, they cannot receive AB blood and they cannot receive B blood. People that are blood type B can receive blood from other B's or O's, but they cannot receive blood from A's or AB's. People that are AB can receive blood from A's, B's, and O's. They're called the universal recipient. Whereas people that are blood type O, as I showed you, can only receive from other people that have blood type O. Here you see what I was trying to draw on that whiteboard there, the idea of the A and B and AB antigens. Type O is just the absence of A and B antigens. So people that have blood type O are going to produce anti-A and anti-B antibodies, whereas people with blood type AB don't produce any antibodies, which is why they're the universal recipient. There's a condition called hemolytic disease of the newborn. This occurs during the second pregnancy of a woman who is Rh negative. So, if a woman who's Rh negative gets pregnant with a baby that is Rh positive, and that can be due to the father being Rh positive, that baby, when it's born, the placenta and uterine lining actually exchange blood briefly at birth. In that case, the woman's body will recognize those antigens and create anti-Rh antibodies. So those anti-Rh antibodies build up over time, and if that Rh negative mother gets pregnant again with another Rh positive fetus, her antibodies can attack the fetus and cause a disorder called a blue baby or hemolytic disease of the newborn. It could potentially kill the fetus. So women who are Rh negative, who have a chance of having an Rh positive baby for the second time, will go on a substance called RhoGAM, which basically desensitizes their body to that Rh antigen, so their baby can live healthily. Blood chemical composition tests are going to use the plasma or serum that is extracted to determine various characteristics. The plasma is the liquid portion of the blood, so you'll need to have anticoagulants in the collection tube to remain in liquid form, whereas the serum is the plasma without the clotting factors. It's the liquid portion that remains after the clotting factors have been taken out. So some blood conditions include polycythemia. This is where you have too many red blood cells. Polycythemia vera would be just too many formed elements, so not just red blood cells, this would include white blood cells. Anemia would be the opposite, too few red blood cells, or deficiency of functional hemoglobin. There's also leukemia, which is red bone marrow cancers, which leads to abnormal blood cells. And then other infections, including bacterial, fungal, viral, or other parasitic infections, can influence our blood composition. Anemia can be due to low red blood cell numbers. There's pernicious anemia, which is insufficient hemopoesis or hematopoiesis. In other words, your red blood cells aren't forming properly. Hemorrhagic anemia, that's caused by excessive blood loss. This can happen to some women on their monthly cycle. There's also hemolytic anemia, which is where you have many ruptured red blood cells, or aplastic anemia, where the red bone marrow has been destructed due to toxins, radiation, and medication. Therefore, red blood cells are not being formed as they need to be. There's anemia due to hemoglobin deficiency, like iron deficiency anemia, inadequate intake or absorption of iron from the digestive tract, or excessive loss of iron through hemorrhaging can cause iron deficiency anemia. There's also sickle cell anemia, which is actually a genetic disorder where the hemoglobin protein is defective. It leads to a sickling shape of the red blood cells instead of being a disc shape, so they do not carry oxygen as effectively. Common blood tests, you may even get to do some of these in lab, are the hematocrit tests. The hematocrit test is telling you the overall percentage of red blood cells, so this could help diagnose anemia or polycythemia or even dehydration because you can see the plasma levels. The average or normal values, hematocrit values in females, are lower than in males, so 38 to 42% hematocrit in females, whereas 40 to 54% in males. If you have a high hematocrit, that indicates polycythemia, whereas a low hematocrit would indicate anemia. There's a reticulocyte test. This is testing the percentage of reticulocytes, so this is assessing the rate of red blood cell formation. This could indicate bleeding or hemolysis or iron deficiency or even a malfunctioning with the red bone marrow, nutritional deficiency, or leukemia. There's also a differential white blood cell count. This is going to give you a percentage of each type of white blood cell in a sample of 100. So this is going to assess immune status. So if you're having some type of issue in your body, your doctor may issue blood work, which includes a white blood cell count, where they'll actually get your blood drawn, and then someone will sit in a lab and count your number of neutrophils, eosinophils, basophils, lymphocytes, and monocytes. And the various numbers of those could indicate different maladies in your body. There's also a CBC, or complete blood cell count, which gives your overall red blood cell count, your hemoglobin count, your white blood cell count, your platelets, your hematocrits, and your differential white blood cell count. So it could break down all of your blood cells. And that ends chapter 10.